few weeks ago, I went to Niagara Falls with some old friends. And it was super fun because I hadn't seen them in a long while. But compared to past trips, the mood was a bit different. And this was because the father recently had been diagnosed with a rare stage, late stage liver cancer. And the mood was different for the whole trip, but this really hit me at the last dinner because we're sitting down eating food. It was a nice Brazilian place, and in the middle of the dinner, their father looked over to me, and he knew that I did some work with immunotherapy, and he asked me the question, how much longer can I live? And in that moment, I really realized that this is the question that all patients want the answer to. We can talk about innovative treatments, innovative therapies, innovative diagnostic techniques, but at the end of the day, a patient wants to know, can I see my kids graduate, or can I go on one last family vacation? And despite the heaviness of this question, it's not that got to me. It's the fact that I didn't know the answer. Not because I'm young and I'm not supposed to think about these sort of questions at this kind of age, but because no one knows the answer to this question. We have new therapies coming out every day. We have immunotherapy for cancer. We have stem cell therapy for a whole range of aging-related diseases. We have gene therapy and gene editing for a multitude of diseases. But we know that they kind of work for most patients, and we don't really know why they work, or we can't tell a patient if they're going to survive or not, they're going to have a good or a bad response. And that annoyed me. So over the summer, continuing this year, I've been trying to answer that question for cancer immunotherapy. So I worked with the Princess Margaret Cancer Research Tower, and we took 10 patients. And we gave them a type of immunotherapy where we take their T cells, their cancer-targeting T cells out of them, we expand them, so we clone them, make more of them, and then input this larger amount of cancer-targeting T cells into their body. And then after this infusion, every 2 to 3 months after, we take blood samples from them. And our goal for this, when taking the blood samples, is trying to define the specific set of T cell receptors the patient has in their blood. And the T cell receptor is the actual part of the T cell, which is going to go out to a specific cancer cell, bind to it, and elicit the immune response. And T cell receptors are super specific. You can kind of think of them as a key, and each key will have a specific keyhole that it's looking to target. And by taking these blood samples, we're hoping to get the information about the set of keys a patient has in their body, and hopefully answer the question, how much longer can patients live? Are they having a good response? Why are they having a good response? Why are they having a bad response? So from this blood sample, we isolate all the T cells in their blood, and from the T cells, we isolate the DNA and the RNA. And we know that a specific small subset will be relevant to tell us about the specific T cell receptors that they have. Now, it was a lot of fun doing this, and we learned a lot about patients and how they respond to immunotherapy, but I don't think that this process will allow us to answer the question, how much longer can a patient live? And the reason for this is it's extremely laborious. We have to take the blood sample, isolate the T cells from it, isolate the DNA and RNA from the T cells, isolate the specific small percentage of the genes that we want in the T cells, and then analyze it and hope to get a good response out of it and get a good analysis. And for this, I wanted to improve it, so I asked, okay, let's do circulating free DNA as well. And circulating free DNA is just circulating DNA around your bloodstream. And when we take this blood sample out, we know that just like the DNA and RNA, a small percentage of this circulating free DNA will be able to tell us about the T cell receptor. Now, this new technology falls under the exciting new realm of liquid biopsy. There's been a lot of hype around liquid biopsy. They have an 88% stage 4 cancer diagnosis, but there's still major problems. We're not getting enough information out of this circulating free DNA. And we do have a high diagnostic rate for stage 4 cancer, but it's not a miracle of science that we're diagnosing stage 4 cancer. It's a miracle to diagnose stage 1 cancer with a blood sample, and that diagnostic rate is 32%. And for my study and for other studies, we can't accurately track the response to treatments with circulating free DNA. I think of it like, if we're trying to find a specific gene in a blood sample using liquid biopsy, it's kind of the same as trying to catch a fish with a ladle and a bucket. And You know, if you put a bucket down into the ocean or a lake and you pick it up and you expect there to be a fish there, you're ignorant. But we do the same thing every day with patients. And you know, if you really wanted to catch a fish, you would use a fishing rod, right? And we don't have that same technology to find certain genes in circulating free DNA. So I set out on the journey to find a fishing rod for circulating free DNA. So what really makes a fishing rod great? What makes it 10x than a bucket, right? And there's three main things. The first one is prolonged exposure, so there's a greater chance of finding a fish. The next one is a capture device. So there's a hook so you can capture in and stay on to the fish. The last, and probably the most important, is a selective capture bait. You know, corn or a worm or a plastic ball that allows fish to be attracted to your hook. And we need all three of these in order to capture the specific circulating free DNA we want from the blood. So the easiest one to get by is the prolonged exposure. You know, if we have a peripheral venous catheter, a patient will come in, and in 10 to 30 minutes, this catheter will be entered in, and it'll capture all the relevant pieces of circulating free DNA that we want. And then after this, we'll be able to find and analyze the patient's blood. Now, the hardest part here is now the selective capture and the selective bait. Now, in order, like any good fisherman, in order to find what fish you want to catch, you need to learn more about the fish. Now, the T-cell receptor is separated into two parts. There's an alpha chain and a beta chain. These two parts combine to form the T-cell receptor. These two chains come from two different genes, and these genes are what we want to capture. These genes are what I was looking for when isolating from T-cells. So these genes, the alpha and the beta gene, create the alpha and the beta chain, which then combine to form the T-cell receptor. That'll go out and elicit the immune response. Now, just like a fish, in order to tell that it's a fish, you have different parts, right? You have, okay, it has a mouth, it has a fin, it has a tail, and it's a bunch of scales. And that allows you to know that that's a fish. And just like a fish, our genes have multiple parts, and these are called V, D, and J. The names are not super important, but just realize that there's three main parts, V, D, and J. And there's also a C part, but that doesn't tell us specific information about the T-cell receptor. So we'll need to capture this gene in order to have a good analysis of the blood sample. So in my project, we kind of have this bait technology called CAP-TCR. And what CAP-TCR does is it chucks out baits to a large sample of DNA, and it can bind to these certain V, D, and J segments specifically, and then it'll take the whole gene out so we can learn about the T-cell receptor. We just need this technology to work inside of our bloodstream actively to capture the circulating free DNA. And in order to do this, we need a strong and biocompatible capture device, just like a hook. And what this comes with is carbon nanotubes. So carbon nanotubes have been known to be very strong and very biocompatible, but we're also able to manipulate them. So we're allowed to create large forests of vertically aligned carbon nanotubes. So let's say we take a wall of carbon nanotubes, and what we're allowed to do with them is we're allowed to functionalize them. So you're allowed to wrap certain molecules around them. In our case, it's DNA. And when we wrap DNA around it, the great thing is that when we pass corresponding strands of DNA, then this DNA will bind to the DNA on the carbon nanotubes, and we're allowed to selectively capture whatever piece of DNA that we want. Now, if we replace these strands of DNA with the strands of DNA that we have in CAP-TCR, we're allowed to selectively capture these specific pieces. And if we put this on the end of our peripheral venous catheter, we enter it for 10 to 30 minutes, so circulating free DNA will pass by, and we're allowed to get an actually good analysis of our T-cell receptors. And if a technology like this could work, the impact of it spans much wider than just tracking immunotherapy, no matter how great that is. We can get that mere 32% diagnostic rate up, and we can find rare mutations in cancer and be able to predict other responses more accurately. And ultimately, we can finally answer the question, how much longer can a patient live? Thank you.